The present now in the screen is Edgar, Vani, and me, and we will allow to Alexei to start with the school and the initial presentation. Thank you.
And before, just one couple of words, just to say to everybody, thanks a lot for being here. And we hope that this school will fulfill your expectations, and I'm sure it will be like that. And as Jose Ramon said, for now, the one who made the introductory remarks on the first talk will be Alexei Verkhratsky. And he's a professor of neurophysiology at the University of Manchester. His main line of research is about the physiology and pathophysiology of neuroglia, calcium signaling, and brain aging. Of course, he's a member of many different academic societies, and he even has a Wikipedia webpage, which is very nice. And well, so we can go straight to listen to Alexei. Alexei, thanks very much also for this talk. Thank you.
Good morning, everybody. And of course, before I start my lecture, ciao, Elena. Before I start my lecture, I probably will say a couple of words about a neuroglial school, about history of Astroglia, about our mission, about our aims, and about our future. So Astroglia Center of Neuroscience was probably the very first. Elena, good morning.
Good morning.
Probably the very first neuroglial center in the world. And even now there are very few glial centers in the world, and that is very, very strange, because somehow during last 100 years, glial cells have not been in the focus of neuroscience, which was completely different if you compare it to the end of 19th century, because the concept of neuroglia and the word glia appeared 50 years before the word neuron appeared. And in the second half of 19th century, practically every neuropathologist, every neuroanatomist was engaged with glia. You can't imagine, I mean, there is not a single prominent neurophysiologist, neuropathologist, neuroanatomist of 19th century who did not pay any attention to glial research. And many different things have been found. And the most important thing which has been detected at the end of 19th century is the fact that glial cells are contributing to pathology. And they're contributing to pathology in a very peculiar way. They change their morphological appearances. So already by the end of 19th century, it was very clear that there are many cells which are changing their morphology in pathological conditions. And all these cells appear to be glial cells. And that is something which probably would be a very important part of this school, because we are going to talk a lot about pathophysiology of glia, and we are going to represent you a very, very important concept that whenever pathology comes, it's not neurons who are protecting the brain or the spinal cord or the peripheral nervous system. These are neuroglial cells, and which are many, which are, of course, as you know, there are astrocytes, oligodendrocytes, oligodendroglial precursors, and there are microglial cells. And one of their absolutely fundamental tasks is to defend the brain. And if they do so, then the pathology is compensated and we can survive. But if they fail, then pathology develops and, of course, neurological outcome comes out. So that was more or less considerations behind the ideas of establishing neuroglial center in Spain, and that happened at the very late 2010s, at the end of first decade of this century. And after several years of quite an effort, this particular center was established. And at the beginning, it only combined several groups which have been in Bilbao working with neuroglia, but during this decade, it grew up very, very substantially. And for the time being, this is one of the very prominent neuroglial center research in the world, which are looking for very many different things, which are specializing on every type of glia, on physiology of glia, and on pathophysiology of glia. And one of the missions of this center was, of course, to propagate the knowledge about glial cells. And that's how we came to this idea of making glial school. And the first glial school was 3 years ago. And our aim at the beginning was to make it regular and to go for these schools every 2 years. Unfortunately, this COVID pandemic, of course, modified our plans and we had to postpone all things. But then we decided not to postpone any longer, and we decided to make this online course. And of course, we understand the limitations of it. At the same time, we do understand that this particular format does have certain beneficial effects. For example, every lecture which will be delivered during this week is going to be recorded. And it means that at your leisure, you can always come to this lecture, you can look into it again, and you can then ask the questions. And I think another very important profit which can be gained from this online format is interactions. And of course, all know our emails, and every student knows the email of lecturers, and you can always send us your questions by email. And of course, we are going to have the brainstorming groups, which is going to be all next week. But this is even beyond that. So if you have questions, you can ask the lecturer either during the session or outside of the session using modern technologies for communications. Now, what is also very important about this school, its school is truly international. And this particular time, we have students from 11 countries, from Mexico, from Latin America to Asia. And I think this is another very important issue which is linked to this globalization of the brain, that our science is becoming worldwide. And fortunately, this technology allows us, even in these times of peril and these times of stress, not to observe any borders, between countries, not to observe borders between people, and we can all come together and enjoy learning new things about neuroglial cells. So with that, I probably will come to an end of my very short presentation. The only thing I would like to say is that all credits for making this school goes to organizers, goes to Vanya, goes to Jose, goes to organizing team, and they did an incredible job in order that this thing will go smoothly and it will be useful and helpful. And so again, welcome to this second Astroglia Neuroglial School, and probably without any further words, I have to start from the first lecture, which, as you know, it will be me, and I will be talking about neuropathology of glia, about many different things, and regard this lecture as really an introduction, because today we have a full astrocytic day, and we have 3 absolutely wonderful leading speakers in the world, and each of them will talk about certain aspects of neuroglial pathology. So I'm going to just introduce the whole thing, and then of course they are going to do the job in order to give you the detailed knowledge. So now we are coming again to the main problem, how to share the file. And share application screen. Probably is this one. Nope. I did it already, but okay, yeah. Do you see it? It should say principles of Astroglia pathology.
Okay, it's fine.
Fine, yeah. So everybody sees it, no problem. If anything, again, this is not a formal meeting. If you need something, we have chat, we have microphone, interrupt me, send whatever messages to us, because we really want the whole thing to go smoothly. So as I said, I'm going to start from a very introductory lecture, in which I'm going to outline the main principles of Astroglia pathology. And indeed, this is a very wide field, and it involves many different changes which are linked to pathology, which can drive to pathology, and which do define neurological outcome of many forms of neuropathology. And that includes everything from reactivity to atrophy and degeneration, as you can judge from this title. So first of all, we are talking about human brain. And human brain is most certainly the most complex system which has ever been encountered by natural sciences. So just to give you an idea, within 1.5 liters of our brain, nature packed something in a range of 200 billion cells, 200 billion different cells, neural and non-neural. How it compares with the rest of universe, it's about the same number of stars in the Milky Way. So indeed, brain has a galactic proportion. And all these 200 billion cells are connected with many, many, many trillions. We don't know how many, most likely more than 100, many, many trillions of connections. And that makes the brain one of the most powerful computing machine in the world and in the universe. Brain has a memory capacity of random access memory in a range of about 2.5 petabytes, and it executes whatever calculations in a petaflop range, which is absolutely comparable with the largest supercomputers which has been so far built by mankind. However, our brain does so within the volume, as I already mentioned, of 1.5 liters, and it does so at the expense of 12 watts per hour. If you go to supercomputers, the supercomputers occupy hundreds and hundreds and hundreds of cubic meters of space, and they need a nuclear power plant nearby because they need tens and tens of megawatts of power per hour in order to proceed. And in that particular respect, when people talk to me about artificial intelligence and when computers will replace humans, I think it's laughable. So mankind so far didn't make anything even close to a computing power and learning power and plastic power which is exerted by the brain. Brain could be regarded as a combination of many different organ-wide networks. And number one, of course, it's connectome. It's all these billions of axons which are connecting these billions of neurons and which really bestow the computing power on the brain, because without those connections, that would be just a loose organization of many, many, many different cells. Nonetheless, this connectome and all these cells, they obviously need an energy. And an energy for biological system is glucose and oxygen. And that is all delivered by vasculature. And we have an incredibly refined vascular network in the brain. So the total length of vessels inside our brain is probably around 600 kilometers. And we have a fluid system, which is incredibly important too, because first it provides the environment in which cells are also talking to each other through volume transmission. But even more important, it provides this water cover in which brain is floating. And that, first of all, reduces the mass of the brain from 1.5 kilograms to about 40 grams because of obvious Archimedes powers and forces. But second, it allows us to move and think, because if this fluid would not exist, of course, every movement would trigger a concussion of the brain, which will hit the skull. So because of this fluid system, we can do what we are doing in our life. And all these brain wide networks are supported by a specific type of the cells in the brain. It's a homeostatic arm of the central nervous system, which of course is represented by neuroglia. So that is a very general classification of what neuroglia is. So are, because neuroglia, remember that it's always plural, of course. So what neuroglia are? They are homeostatic and defensive cells of the nervous system. And neuroglia are generally subdivided into neuroglia of peripheral nervous system and central nervous system. And on top of that, there is a specific group of glial cells which are part of specialized sensory organs, which I think it's somewhat artificial to put them in a separate group, but from the point of view of physiological description, it's easier to put it all in the context. So in the peripheral nervous system, there are, of course, many Schwann cells, and we continue to learn and discover new types of those cells. So, for example, about 2 years ago, a new type of nociceptive Schwann cell has been discovered. There are satellite glia, which are part of peripheral ganglia and which are taking care of homeostasis of peripheral ganglia. And there is a big realm of enteric glia, which sits in the gut and which underlies the proper function of our intestines. In the central nervous system, all glia is divided again into two different classes. One is microglia, which are not cells of non-neural origin, which are invaders, which are mesodermal cells, which are coming to the brain early in development and provide for innate and defensive nervous system part. And there is macroglia of ectodermal origin, so they are signs of the very same neuroepithelial cells as neurons are, and these are represented by a big class of astroglia and an oligodendroglia, which are again presented by myelinating oligodendrocytes and their precursors, also known as Ng2 glia. Now, let's come to neurological diseases. And unfortunately, I have to start with this rather sad statement, because although medicine made a tremendous progress over the last 100 years, and we now know how to cure many diseases, how to change livers and hearts and joints and so on and so forth, at the moment we come to the diseases of the nervous system, we are more or less helpless. We can ameliorate major symptoms, but we cannot cure the disease, mostly because we don't know what is the pathogenesis of those diseases and which mechanisms are underlying pathological progression. And probably another issue in all this particular problem is that for the last 100 years, all neurological thoughts were focused on neurons, on neuronal receptors, on neuronal molecules, with very little attention paid to a neuroglia. And of course, brain is a cellular network which is composed from many cells, and these cells are working together. And I think it's really, really important to understand that all cells in the central nervous systems are created equal. They work in concert. So there are no good cells, bad cells, supportive cells, secondary cells, and so on and so forth. Paraphrasing John Donne, we can say that no cell is an island, entire of it is a piece of the continent and a part of the main. When pathology comes, these cells respond to pathology in different ways. Neurons become stressed, they start to malfunction, sometimes they die, sometimes they survive, but they cannot actively oppose a pathological process. In contrast, what happens to glia, and that, as I said, is known for more than 100 years, glial cells are actively responding to pathology by changing their morphology, by changing their biochemistry, gene expression, physiology, with a major aim of fighting this particular pathological process. And again, if they succeed, then of course they compensate the disease. If we try to make some definitions, so what is disease? Disease could be defined as a homeostatic failure, that is inability to maintain physiological equilibrium. And if this is the case, then obviously we have to look into these homeostatic cells of the nervous system, and we have to conclude that neuroglial failure is a universal pathogenic step in all neurological diseases. And this particular notion was brought forward by the very inventor of the concept of neuroglia, by Rudolf Virchow, who did so in 1858, and he was 37 years old, and he was arguably the most prominent pathophysiologist of 19th century. And so in his lecture to students of Charité, when he introduced neuroglial concept, he said that this very interstitial tissue of the brain and spinal marrow, that is neuroglia, is one of the most frequent seats of morbid change. And that was very much endorsed by, as I said, most of neurologists and neuroanatomists of 19th century. So let's start from the beginning. Astroglia, definition, appearance, function. So what is astroglia? The actual term astrocyte means star-like cell. And appearance of this term is very much linked to Camillo Golgi, who was the first to open the brain, who developed the first true staining technique, which is called black reaction or reazione nera. And when this particular technique was applied by himself to the brain, there were many, many cells, like you can see here, which do have some sort of star-like appearance. What is interesting for Golgi is that he also realized that many of those star-appearing cells are sending processes to brain vessels, and then they form a specific portion which is called end feet, and those end feet are covering brain vessels, and Golgi was the first to suggest that they form an interface between the circulation and brain parenchyma. So what they do, they take cells, things from blood, they distribute it through the parenchyma, and vice versa, they take things from neurons, whatever waste or whatever things which appears in parenchyma, and send it back to the blood. And by using this particular technology, many people already saw these star-like cells, and many different terms, including word star, has been proposed. But in 1895, Mikhail von Lenosek, who is of Hungarian origin, but he was working in Bern at those days, he published a book in which he made a definition of what is astrocyte, star-like cell. He had, however, a sort of prophetic soul, because what he wrote, he said, I would suggest that all supporting cells be named spongiocytes, and only the subpopulation of them be named astrocytes. And that is indeed true, because those of you who worked with astrocytes know that in real conditions in the brain, they very rarely, if at all, do have star-like appearance. They really look like bushes or sponges. And more or less at the same time, the words protoplasmic and fibrous appeared, and people start to appreciate heterogeneity of different morphological shapes of glial cells. Now, astrocytes are actually only a part of a big class of cells, which we know as astroglia. Now, these astroglial cells account for about 20 to 40% of glia. And I beseech you, whenever you start your papers on astrocytes, don't say that they are most numerous cells in the brain or most numerous glia in the brain. This is not true. But this class of astroglia includes very many different cells, very different in their morphology and very different in their function. What unifies them is their origin. So these are cells of neuroepithelial origin, which are responsible for homeostatic support of the nervous system. And the difference could be immense, and as you can see here, for example, ependymocytes or choroid plexus cells, so cells which are having cilia and which look like bricks, and they are lining ventricles and making different barriers and sitting near meninges, they also belong to the class of cells known as astroglia. There are many radial astroglial cells or radial astrocytes, because two radial glia exist only in embryogenesis. And again, they have a very specific morphology, but their function is always the same. It's homeostatic support and defense. And of course, astrocytes are the most prominent part of this group of cells of astroglia. And again, there are many different types of astrocytes. Some of them are mentioned here. Some of them probably are going to be discovered in coming years, because every now and then we are seeing a new subtype of astrocytes being described. What is a definition? Astrocytes are homeostatic cells in the central nervous system, and they support homeostasis of the CNS at all levels of organization, starting from molecular homeostasis, because they are responsible for ionostasis, for pH regulation, for water transport, and very importantly, for homeostasis of neurotransmitters. They're supporting cellular and network homeostasis, because astrocytes are stem cells in the adult brain, and they are indispensable for synaptogenesis. They provide neurons with metabolic substrates. They support organ homeostasis by maintaining blood-brain barrier and by maintaining operation of the lymphatic system. And they are contributing to systemic homeostasis being a hemosensing cells of the central nervous system. How to study and visualize astrocytes, and this is a task very far from trivial, because so far we do not have a universal for astrocytes. For a long period of time, such universal marker was considered to be glial fibrillary acidic protein, and indeed in vitro research shows that up to 95% of astrocytes in culture usually express GFAP at the level of detection. In real brains, this is not the case, and in healthy brain, in many regions, maybe 10 to 20% of astrocytes do express GFAP at the level of detection. Another problem with GFAP is that it's not a faithful morphological marker because GFAP stains only the cytoskeleton, and it doesn't stain a main part of astrocytes, which is represented by peripheral processes. So to use different techniques for visualizing astrocytes, like different markers, for example, here you can see some examples of staining with antibodies against protein S100 beta or glutamine synthetase. And now you see the picture is very different because we not only see major processes, but we also see some smaller structures which are reflecting peripheral processes of astrocytes. And if these astrocytes would be stained intracellularly with fluorescent dyes or by expression of fluorescent proteins, then of course the picture will be very, very different. And that will show you this bush form, spongiform appearance of astrocytes, which is reflected their specific morphology. So more than 80% of surface area of astrocyte belongs to peripheral processes. And those are peculiar structures. They are very thin. Usual thickness is about 100 nanometers, and they are covering synapses, and they are covering really, really big number of synapses. So a single human protoplasmic astrocyte covers up to 2 million of synapses, obviously providing them with homeostatic support. And that's how it looks like for many, many synapses. We still have certain problems in understanding how this structure is made. But nonetheless, about 60% of excitatory, mostly synapses in the central nervous system, are very tightly covered by astrocytic membrane. And these perisynaptic structures, as I said, are incredibly thin, but they are incredibly important because they contribute to all stages of life of the synapse, from synaptogenesis to maturation, isolation, maintenance, and even extinction, because astrocytes are labeling redundant or non-needed synapses with certain tags, which allow microglial processes to come and to eat them away. And homeostatic support is maintained by an incredible number of transporters expressed in astrocytic membrane. And again, the surface of this membrane is very high, so we can put many processes which are responsible for all aspects of homeostatic control. Again, control of ions, control of neurotransmission, supply of neurons with neurotransmission precursors, and so on and so forth, and so on and so forth. On top of that, of course, astrocytes are secretory cells, and they secrete up to 200 different molecules of all these different subtypes, transmitters, modulators, plastic factors, you name it. And this secretion is done by 3 complementary pathways: by exocytosis, by diffusion through the channel, and by action of transmembrane transporters. And astrocytes have many secretory organelles of different size and function and cargo and so on and so forth. But what is important, the exocytosis in astrocytes is very different from neuronal exocytosis. And the reason is a differential expression of one of the proteins of exocytotic machinery, which is SNAP25 in neurons, SNAP23 in astrocytes. And that confers an incredibly fast exocytosis in presynaptic terminal, which underlines a synaptic transmission, and very slow, hundreds of seconds, minutes, or even hours secretion from astrocytes, which are not aimed at a fast transmission, but rather aimed at neuromodulation and signaling through extrasynaptic pathways, which is known as volume transmission. So after this brief introduction into astrocytes, let me go to Astroglia pathology. And again, we are still in the nascent state here. We are still only starting to learn. And I'm sure that this classification I'm showing you is going to be modified very much within the next decade. Nonetheless, we can distinguish 3 major groups of pathological changes in astrocytes. First is degeneration. Of course, astrocytes are cells, and when they are hit hard, they die. And that happens in stroke, that happens in aging, that happens in severe trauma. And morphologically, this degeneration sometimes is manifested in the form of something which is called plasmatodendrosis. The word was introduced by Ramonica how many years ago, and that is actually fragmentation and bedding of astrocytic processes. This is death. And of course, when astrocytes die, homeostatic support suffers very much. The second big part is reactive astrogliosis, of which I think Elena and Milos are going to talk much more, so I'm going to just introduce it. And reactive astrogliosis is something which has been seen by neuroanatomists 100 years ago. It's a morphological and functional remodeling of astrocytes. And this remodeling produces many different reactive phenotypes. And again, our classification is so far very simple and oversimplified and primitive. We can classify them according to morphology, according to severity, protection, scar forming, you name it. But to be honest, we still don't know all the extent of phenotypes, of reactive phenotypes of astrocytes in the context of neurological diseases. And finally, there is a big class of astrocytic pathological changes, which is generally known as astrocytopathies. And these could be, again, very roughly subclassified into pathological remodeling, in which astrocytes acquire new features which drive the disease, and atrophy and loss of function, in which astrocytes become smaller, they become morphologically atrophic, and they lose their function, so they reduce homeostatic support, from which obviously brain tissue does suffer. So again, a couple of words on reactive astrogliosis, and I'm sure that Elena, who is going to talk immediately after me, and Milos, who is going to talk today at the end of our session, are going to really elaborate on that, because it's one of the cornerstones of glial pathology, of course. So historically, astrogliosis was defined already at the beginning of 20th century, and it was defined as hyperplasia and hypertrophy of astrocytes associated with fibrous change, that is, with scar formation. So now we know much more, and I'm very happy and proud that I was part of the team which made this consensus story on reactive astrogliosis, which was published recently in Nature. And again, some of co-authors are our speakers today. And that defines reactive astrogliosis is an evolutionary conserved, primarily defensive response of astrocytes to pathology, which produces many phenotypes which are age, disease, context specific. And of course, you all heard about this ideas of good astrocytes, bad astrocytes, A1, A2, all these concepts are highly misleading. We don't have black and white in nature quite often, so there are many different reactive astrocytes. What triggers astrogliosis? Many different factors of different origins. They can come from the cells, from microglia, from synapses, from neurons, or they can be blood-borne when blood-brain barrier becomes compromised and these factors go to parenchyma and they instigate many intracellular signaling cascades which trigger astrogliotic remodeling. And after astrocyte is remodeled and becomes reactive, it also starts to secrete many things which are generally aimed at reparation and regeneration of, for example, repair of blood-brain barrier, or promoting post-lesion regeneration of neuronal networks. Again, as I said, there are some applications based on morphology, like isomorphic, no change in territorial domain, astrocyte hypertrophic, but they don't move anywhere. After resolution of pathology, they go back to healthy state, or an isomorphic in which astrocytes move. Sometimes they start proliferate. By the way, this is not very often, and that results in formation of glial scar. Or it could be moderate or severe. And even if you think about glial scar is still also a pure defensive reaction, because what it tries to do, it tries to undamaged part and save the whole at the expense of the part. Pathological remodeling, just give you an example, and one of the most illustrious example is, of course, Alexander disease, in which astrocytes express sporadically mutated glial fibrillary acidic protein gene, which changes clearly their cytoskeleton, and that results in accumulation of extracellular protein depositions around vessels in astroglial end feet, which are called Rosenthal fibers. But the real outcome is a severe leukomalakia, its disappearance of white matter. And how purely astrocytic changes result in the appearance of white matter, we still don't know, but we also know that astrocytes are instrumental for many other white matter pathologies and for many other leukomalakias. Again, what are the mechanisms remain to be elucidated. Another example of pathological remodeling, it's another primary astrogliopathy, it's hepatic encephalopathy, in which follows the liver failure. And when liver fails, for example, mushroom poisoning, then of course ammonia goes up in the blood, ammonia goes up in the brain, and the major detoxifier of ammonia is glutamine synthetase. It's an enzyme which in the CNS is exclusively sitting in astrocytes, and this enzyme is absolutely and indispensable for produces glutamine, the main and obligatory precursor for both neurotransmitters. And when ammonia is too high, this particular pathway is clogged, and that changes astrocytes fail to maintain potassium homeostasis, and they downregulate glutamate transporters, and they start to produce aberrant calcium signals with aberrant secretion, so they start to drive the disease, and clinical manifestation is acute psychosis. And that is really important from clinical point of view, because quite often actually these patients have been driven to psychiatric ward when of course they died because they never etiological cure. Astrocytic atrophy in diseases started to be appreciated for probably maybe last 10, 12 years, and it all started from psychiatric disorders and most specifically from depression. Of course, it's very difficult to model psychiatric disorders in animals, and it's even more difficult to study humans, but nonetheless, one of making animal depressed is a chronic unpredictable stress. And how can we know that animal is depressed? And there are, of course, many behavioral tests, but one test is very simple, and that is test for sucrose preferences. So in normal conditions, sweet water. So they consume more sweet water than normal water if you have both sources. When they become depressed, they stop liking sweet water. And that is very similar to one of the leading symptoms of depression, which is called anhedonia. That is inability to derive pleasure from pleasurable things. It's easy to see, and it's very remarkable. So what happens to astrocytes when animal is stressed? I think that was a seminal experiment made, as I said, about 30 years ago by Banasser and Duman. And what they did, they stressed animals, they watched their sucrose preferences and other tests, so they ensure that the animal is stressed and depressed, and then they looked for astrocytes. And there are much less astrocytes, and those astrocytes which are there are much less complex. And then they made the second step. So what they did, they tried to selectively kill astrocytes in prefrontal cortex, Astroglia toxin. That killed astrocyte, and that by its own self triggered depressive behavior. And when they injected neurotoxin and killed neurons, this behavior was not there. So that was the very first demonstration that astrocytes are really incredibly important for controlling the mood. And that is really the atrophy of these astrocytes or their death, which is underlying particular pathology. What happens in humans, and these are coming from Grażyna Rajkowska, who spent many, many years studying astrocytes in psychiatric diseases, and these are astrocytes in white matter of severely depressed patient, and of course this is post-mortem study. And as you can see, the number of astrocytes is decreased absolutely dramatically, like 95%. And those astrocytes which are remaining, they have much smaller area fraction of GFAP profile. Schizophrenia. These are now experiments made by Mike Nedergaard and Steve Goldman. And what they did, they took peripheral cells from boys with early schizophrenia, they made stem cells, and they made astrocytic precursors, and grafted them into the rodent brain, and then compared them. And as you can see here, it's very, very clear. Astrocytes differentiated from cells from diseased patients are much smaller and much less complex. Cocaine addiction. Again, the very same thing. So you can make poor mice cocaine addicted. And when then you start to look into astrocytes in nucleus accumbens, which is, of course, a relevant area for this pathology, astrocytes become smaller and much less complex. Epilepsy, and that is really interesting because in epilepsy we know that we are dealing with a severe astrogliosis. So really in epilepsy, astrocytes upregulate GFAP and they become very hypertrophic from the point of view of GFAP images. But when these cells are now morphologically assessed and their complexity is assessed, as you can see here in epilepsy, there are much less processes, much less synaptic coverage. So again, these cells become atrophic, even though they have an appearance of reactive cells. So we can have reactive astrocytes, which an atrophic peripheral morphology. Parkinson's disease, again, stem cell technology and comparison of healthy controls with people from Parkinson's disease. And this particular study was done in Bilbao, in Achucarro, with Fabio Cavaliere and Carlos Matute and other colleagues. And as you can see here, it's exactly the same. Astrocytes derived from stem cells from patients with Parkinson's disease are smaller and they have much less complexity. So indeed, this is an atrophy which is present in many, many different diseases, especially of diseases connected to control of mood and cognition. And now I have last 20 minutes of my talk, and I go to Alzheimer's disease, which is indeed sort of a scourge of mankind, because now we are facing a pandemic of Alzheimer's disease and senile dementia, with numbers increasing every year. And here you can see Alois Alzheimer and his patient, Augusta Detter, whom he saw in 1901, and she was severely demented with a number of psychotic symptoms. And she died in 1906. He did autopsy and found in the brain senile plaques and intraneuronal tangles, which is hallmark of the disease which bears his name. And the Alzheimer's disease was defined by a boss of Alois Alzheimer, by Emil Kraepelin, who was the most influential psychiatrist of the turn of 20th century. And in 8th edition of his textbook on psychiatry, he made this definition of Alzheimer's disease as a rapidly progressive, young onset, very peculiar dementia with pathology distinct from senile dementia. Because indeed Alzheimer's disease, by definition, as it was revealed from Augusta Detter, was familial Alzheimer's disease or early onset Alzheimer's disease. Now I would like to share with you this very interesting book, which is called Old Age, and which was published in 1889. And I was very lucky to find it somewhere on the stall on the street in London many years ago. And this is a very detailed investigation of different organs and systems of old people. Altogether, they looked for 900 people who were 80 or older, out of whom 74 were centenarians. And what was the conclusion? The conclusion was that the brain sustains aging much better than any other system in the body. So only 2 out of 74 centenarians show problems with memory, were demented, and generally in all these 900 people, memory was good in 78% of them. What we have today in Netherlands, year 2000, 88% of all centenarians were demented. Now again, how to interpret this data? I don't know, it's very, very, very difficult, and there are many different factors. Nonetheless, it seems to me that something did happen over the last 150 years. Something changed a life of Homo sapiens, whether it's environmental factors or social factors or combination, but altogether they brought this pandemic of senile dementia. And if this is the case, it gives us hope, because if we remove it, we can cure this particular condition. So neuropathologically, neurodegeneration, usually treated from neuron-centric view, it's a neuron-centered process which kills neuron, which it obviously does, despite the fact that Alois Alzheimer by himself clearly indicated the role of glia. And here you can see his original drawings of pathological neurons surrounded by glial cell spheres, which are activated or reactive microglia. And here is his own drawing of senile plaque. Again, there is a beta amyloid core, which is surrounded by polymorphic cells, which are again reactive glia. And if we repeat this today with different immune markers and confocal, we will see the same. It's a core and reactive glia around. Now, the main problem of Alzheimer's disease research is that we don't have naturally occurring disease in animals, so we have to resort to models. There are many models, and the current state-of-art models are transgenic models in which these mice overexpress mutant genes isolated from familial Alzheimer patients. And they do represent certain parts of the disease. They produce senile plaques, they produce tau, depending on combination of the genes. And yet there is a big difference between patients and overexpression-based mice. Most importantly, overexpression-based mice do not show massive death of neurons. So neurons are not dying to the extent they die in human brain. These overexpressed mice do have memory problems, that's true, but they never become fully demented. And many processes are very different. For example, very importantly, in the mice, there is a robust neuroinflammatory response, which is not present in sporadic Alzheimer humans. And that, of course, creates quite a misleading ideas about the prevalence of neuroinflammation in that particular disease. Nonetheless, what happens to astrocytes in these animal models? Here you can see, if you look into early stages before plaques, astrocytes are clearly atrophic morphologically, and that is GFAP, but the same could be done with any other marker. But at the moment when to the hippocampus beta amyloid depositions come and form plaques or perivascular deposition, that triggers reactivity of astrocytes. Astrocytes are going to the plaques, surround them, and become reactive. Nevertheless, a part of the plaques, astrocytes remain atrophic. And these atrophic astrocytes are functionally impaired. So, for example, they cannot sustain blood-brain barrier. And this is a rather simple experiment in which endothelial monolayer is grown on the insert, and astrocytes are grown from another site of inserts, and one can measure directly electrical resistance as a measure of integrity of the BBB. And you see healthy astrocytes increase this resistance. Diseased astrocytes cannot do so. Why? Because healthy astrocytes secrete something which upregulates expression of tight junction proteins. Diseased astrocytes cannot do so. But what is even more important, these atrophic astrocytes fail to protect. And this is again hippocampus, and there are depositions of beta amyloid which are surrounded by reactive astrocytes. But if we go to prefrontal cortex or entorhinal cortex, there are no reactive astrocytes. Again, this is, of course, animal model. And what does it mean, no reactive astrocytes? And this is Miloš Pekny, a seminal study done by many years ago, in which he demonstrated that if you suppress astrogliosis by knocking down GFAP and vimentin, then Alzheimer's disease pathology is severely exacerbated because astrocytes don't protect. Now, if you look at temporal progression of the disease, it usually starts in entorhinal cortex, goes to prefrontal cortex, and then comes to hippocampus. And this coincides precisely with an ability of astrocytes to protect. So when atrophic astrocytes cannot mount so this is a sort of stages of Alzheimer's disease, we have atrophy of astrocytes, which reduce homeostatic support, which reduce synaptic coverage, and may be responsible for very early cognitive deficits. But then, of course, disease-specific lesions arrive, that is plaques and tangles and maybe something else which we don't know, and that instigates glial reactivity of both astrocytes and microglia. And that is corresponding to BRCA1 to 4 stages of the disease, which are still compensated. So clinically, it's a mild cognitive impairment. And this mild cognitive impairment can be there till the end of the life of the human beings. So disease could be continued forever. However, if glia becomes paralytic and if glial cells cannot anymore protect the brain, then we have a switch from mild cognitive impairment to dementia and to BRCA5 and 6 stages of the disease, and of course to a fatal pathological outcome. Now, all these are animals. What happens in humans? And I think this is really important because human glia is very different from rodent glia. Now, I always want you to see this slide because still very, very often you can read that there are 10 times more glia than neurons in the human brain. This is wrong. In total, numbers are more or less the same, although there is quite a bit of regional differences within the brain. Now, what is correct, though, is that human astrocytes are much more complex than rodent astrocytes. And here you can see a comparison of mice astrocyte A and E and human astrocytes B and F. And you see how much larger is this astrocyte and how much more complex it is. On top of that, human brain contains certain forms of astrocytes which are not present in any other animals except of higher primates. And one of the most prominent type is a so-called interlaminar astrocyte, which has been seen from the very beginning, and you can see it on historic pictures. So, for example, in Cajal image in the middle, it's the cell which is marked with A. So these astrocytes have small cell bodies immediately under pia and extremely long processes which are crossing several layers. That's why they called interlaminar. Now, what happens in Alzheimer's disease? Here you see a comparison of healthy brain, of Alzheimer's disease brain, same age. Healthy brain has lots and lots of interlaminar processes which completely disappear in Alzheimer's disease. And of course, this is a major challenge because we need to study these cells which are so prominently responding to the disease, and we still have very little opportunity to do so. Now, what happens generally with reactivity of astrocytes in Alzheimer's disease? Now, again, this is Wendy Noble in London who did very simple quantification of GFAP in post-mortem brain, correlating it to the BRCA stage. And you see, at the very early Alzheimer's disease, when the BRCA stages are increasing, 1, 2, 3, 4, GFAP expression goes up, indicative of reactivity of astrocytes. Now, 5 and 6, GFAP goes down, showing the failure of reactivity. Postmortem analysis, different markers. So this is family Alzheimer's disease from Columbia family. And what happens here, again, there is clear decrease in the size of astrocytes in this postmortem tissue. In vivo imaging, this is PET scan made in Nordberg in Karolinska in Stockholm. And what she was doing, she was looking at the signal produced by labeled deprenyl. And deprenyl is a blocker of monoamine oxidase B, which in the brain is mostly sitting in astrocytes. So when deprenyl signal is increasing, that is somehow indicative of increase in astrocytic volume and therefore indicative of reactive astrogliosis. Again, the same thing, initiation of the disease, mild cognitive impairment, and the signal from deprenyl goes up, indicative of reactivity. And then there is a switch, and this is the very same patient. The switch from MCI to Alzheimer's disease to dementia, and that coincides with a big drop in the signal. So again, showing that astrocytes cannot be protective anymore. These are all snapshots. What can we get from sort of more physiological approach? Induced stem cells, a bit of a magic in the dish, and we can produce neurons and astrocytes. And for example, we can produce astrocytes from healthy subject or disease controls from this stem cell using this stem cell technology. These astrocytes have proper morphology, proper markings, and so on and so forth. And now we compare healthy with Alzheimer's disease. And again, what you immediately see, Alzheimer's disease astrocytes are much, much, much smaller. And if we do the very same cultures from neurons and astrocytes from the very same stem cells, again, neuronal cultures look more or less all right. So Alzheimer's disease are probably less dense. But if you look into astrocytes, it's immediately big difference. Healthy astrocytes are severely impaired. Now, can we do something with that? And of course, it's probably almost completely unlikely to make a single pill which will cure Alzheimer's disease. Alzheimer's disease, most likely it's an umbrella term, and there are many different pathological processes which are converging into cognitive impairment. Nonetheless, what we do know is that lifestyle is very important for defining cognitive longevity. And the lifestyle could be, of course, one of the most important, one of the most usual thing. It's a diet, and we all know that caloric restriction is beneficial for prolonging lifespan and for decreasing somehow certain age-associated diseases. So if we expose animals to caloric restriction, what happens? And what happens, astrocytic morphology is increasing, and mostly they increase their very peripheral processes, so they increase the processes which cover the synapses. And what does it mean? That the more processes are covering the synapses, the better are conditions for homeostatic support, so they have better ability to take up glutamate or potassium, and indeed, if we measure directly glutamate transporter currents, we see that in calorically restricted animals, they take more glutamate and do it faster and more effective. And what does it mean for memory and memory-associated processes? It means better neural plasticity, and indeed, caloric restriction increases the plateau phase of long-term potentiation, mostly because of astrocytes which are producing better homeostatic control. What else can be done? Environmental stimulation, which is enriched environment, social engagement, intellectual engagement, and physical activity. And if we take our animals and put them into running, into enriched environment, and again, these are experiments done in Achukara, what we can see is that, first of all, it improves their memory and behavior. But what is most important, it rescues atrophy of their astrocytes. Their astrocytes become more complex and they become larger, and even moreover it rescues impaired neurogenesis. And again, neurogenesis, as you remember, is a function of radial astrocytes sitting in neurogenic niches. So physical activity and enriched environment both can save neurogenesis and therefore save astrocytes. So we are coming to conclusion, and I hope I convinced you that astrocytes are cellular elements for neuropathology. And targeting of astrocytes may provide a new principle for treatment and prevention of neurological conditions. And in particular, a lifestyle is very important for keeping these astrocytes healthy and safe and for prolonging our cognitive longevity. And with that, we are coming to 2000 years before, coming to Marcus Tullius Cicero, who said that senile imbecility doesn't occur in old men, in all old men, but only in those of feeble mind. And we have to fight aging. We must combat it as we fight a disease. A fixed regime, taking exercise, enough food and drink, and be happy. And I hope that you will all follow this healthy lifestyle and prolong your cognitive longevity. And these are many people with whom I was working in glial research, and on their behalf, I thank you for your attention.
Thank you very much, Alexei, for this fantastic presentation. And
just a second,
yes.
Oh, right. I'm back.
Thank you very much. We have already 4 questions from the students. So first, Jorine, Angela, Mar, and Maria. So if you can turn on your cameras if you want to ask directly to Alexei, please.
Milos.
Hello, Jorine, or the students?
Oh, sorry, I'm muted, I see.
Yes.
Hi guys, I'm Jorine Geertsma. I'm from the University of Amsterdam, and I have a question about this GFAP, because of course it's used as a classical marker for astrocyte staining. I'm just wondering, wouldn't you miss out on a certain population if it only stains about 10 to 20 homeostatic glia? And what other markers could you use to try to incorporate more?
That's a wonderful question. And I mean, if you start to look into relatively old morphological studies from 1990s even, this already has been mentioned quite often. So for example, in cortical regions, about 20% of astrocytes were GFAP positive. And if you take S100 beta, for example, it was up to 70%. However, if you start to use different markers, of course, there are many markers, you will have certain overlap, but you still will have certain populations which are stained almost solely with one marker. So in a sense, I think that probably, I mean, we need to use something really non-specific, like, for example, Golgi staining. And it's very interesting because if you do Golgi staining, you mostly stain astrocytes, not neurons. Still, I don't understand why, but that is the case from Mori. What is really important, and it has to be taken in consideration, when we use the expression under GFAP promoter, that labels many, many more cells. Because, of course, all cells do express GFAP. The question is level of expression.
Yeah. And is that then via in situ, or how? So how is that then done under a promoter?
Oh, that's lentiviral, for example, or you do knock-in animals. I mean, there are many technologies. So more or less, you introduce a gene of whatever you want to do under control of GFAP promoter.
Okay, thank you.
Thank you very much, Jolene. So after, if Angela is, you can ask your question also to Alexei.
Yes. Hi, very nice presentation. So the question was related to the astrocyte the pathological astrocytes in these psychiatric diseases. So is it known or there are evidence if for all of them, I mean, the astrocytic pathology is a primary cause, or it is a secondary consequence, for example, of the neuronal damages? So you show for Alzheimer's that this might not be the case, but is this known for those other diseases? I mean, it's a really primary astrocytic failure or secondary damage, let's say?
Well, I mean, of course, it's impossible to answer because clearly for this question, we need to make a longitudinal experiments on human beings. Now, the only thing which is really worth noting that at the moment we start to look into neuropsychiatric diseases, there is absolutely no prevalence of reactivity. So it's, if anything, it's decreasing packing density in astrocytes. This is common for all depression things. There is decrease in number of oligodendrocytes, which is very prominent too. And there is relatively little happening to neurons. So morphologically, glial cells suffer much more. And there is, of course, an immense literature showing that something happens to microglia. Some microglia become reactive, some become dystrophic, whatever it is. But again, here we are entering this particular curse of animal models, because microglia in human brain and in rodent brain with aging behave very differently. So it's very difficult to say. Again, what is the cause, what is an egg, and how happens, that clearly we don't know. But in summary, I mean, in total, all the data are pointing rather to an incapacity of glial support. And if you think about mood disorders as imbalance in neurotransmission, then it makes sense, because all major neurotransmitters are catabolized by astrocytes, noradrenaline including.
Thank you.
Thank you very much, Angela. And then next, Mar, if you can also.
Yes. Can you hear me?
I can hear you.
Fantastic. Thank you very much for your
I can even see you.
Fantastic. I was just wondering regarding this astrocyte morphology that you were showing in all these studies related to neuropsychiatric diseases. Were researchers comparing only the main cytoskeletons of the astrocytes, or also all this peripheral branching that it seems from your talk to me that are so important?
Very, very, very good question. And the answer, unfortunately, is that this so far mostly was confined to GFAP.
Okay.
Now, GFAP can give you some ideas because you can do shoal analysis and things like that and look for intersections. So that somehow gives you an idea of complexity. But of course, it leaves all the peripheral structures out altogether. And that is certainly very much needed.
Of course. Can I briefly just ask you another question, Edgar? May I?
Yeah, sure.
Yeah, fantastic. Thank you. So, well, it's just a curiosity, but as you were showing that in humans, astrocyte morphology is so more complex comparing to animal, for example, and because all these transcriptional studies are kind of showing us that not just astrocytes, but other type of neurons or astrocytes or microglia or oligodendrocytes are different depending on the different brain regions. I was also wondering whether the complexity of astrocytes, whether it has been a study, whether the complexity of astrocytes is different depending on how complex are the functions or how complex are the different brain regions. Do you understand my question?
Yeah, I understand what you mean. Not to my knowledge. I have to admit that there are not that many studies on human astrocytes. Of course, if you want to look into interlaminar astrocytes, you have to look for Jorge Colombo, who started it many years ago and who is #1. When it comes to protoplasmic astrocytes in cortex hippocampus, more or less there are a couple of papers. So I think it's a great idea to look into different regions of human brain and to make a mapping of astrocytes, but I'm afraid it requires quite a bit of an effort.
Yeah,
considering the brain bank is not the best source. So you have to really have a very good postmortem, very good. And a couple of hundred of people with billions of budget who does it serially every day.
Yeah, okay. Thank you very much.
Thank you very much, Mara. So next we have time for two more questions. First, Maria, and then Milos.
Okay.
Maria, are you there?
Hi, yes, I'm here. So I'm Maria from Mexico. So my question is, if GFAP has been, So GFAP has been used as the classic marker for reactive astrocytes, but as you said, it's only detected in the 10 or 20% of the astrocytes. So which and how many markers should be used to talk about reactivation? And I said that considering also the RNA-seq studies.
Many, and I think it's a work in progress, but I'm absolutely certain that Yelena, who is talking right now, will address this question in many more detail. And of course, Milos, who is talking today, will address the very same question from a different angle, but in many more detail. So be patient and wait till the end of the day.
Okay, thank you.
Milos, you are the last question.
Thank you, Alex, for a stimulating and great opening talk. I think many people would dream about the non-invasive opportunity to visualize human reactive astrocytes in conjunction with various neuropathologies. And you have mentioned Deprenil and the work which Agneta Nurberg has been engaged in for a number of years. Could you outline to this audience, because I think there are many people thinking this way, what we can do today with respect to visualizing reactive astrocytes, in particular in human subjects in a non-invasive way, and where should we get tomorrow?
Miller, you are incredibly inventive. I am afraid that in this situation you have to assume responsibility. I don't know, because I'm not working in it. So Atlanta just published this wonderful paper in molecular psychiatry, in which they developed 3 more new astrocytic specific markers for and I know that there are many people in many labs engaged in this for these markers. So far, I think it's just too difficult. Even deprenyl, I mean, we assume that it's astrocytic specific, but this is an assumption.